morning, everyone, dear committee, dear audience. My name is Eszter Gulyás, and I would like to present my PhD topic, which is antimicrobial therapeutic drug monitoring in critically ill patients by a clinical pharmacist. I am a pharmacist working in the university pharmacy department at Semmelweis University, and my vision is to facilitate strong cooperation between clinical pharmacists, clinicians, and the clinical laboratory experts concerning the antibiotic therapy of critically ill. The mission is to promote this antibiotic therapy in intensive care and to become an expert in utilization of these TDM results. I have two ongoing projects, and the specific goals are to investigate the effect of beta-lactam TDM in critically ill patients, and the second is to evaluate the pharmacokinetics-based guidance of colistin therapy. The title of the first project is Beta-lactam TDM is associated with improved clinical cure of the critically ill, but studies with more standardized protocols are needed. And the methodology was used is systematic review and meta-analysis. About the background of this topic is that an estimated 70% of the critically ill patients receives antibiotics while they are admitted to the intensive care unit. And the most frequently prescribed antibiotics are beta-lactams, and the pharmacokinetics of these properties are not stable and hardly to investigate how the pharmacokinetics will be in these unstable patients. So therapeutic drug monitoring is a measurement of the drug concentrations in the blood with a specific interval and to adjust the therapy based on these results. So it's a complex method, and this method could be used for as evidence-based decision-making for healthcare clinicians, especially in the intensive care unit. But the overall impact of this TDM is under intense debate. So our aim was to evaluate the impact of antimicrobial TDM. The question what we phrased was like, is TDM-guided beta-lactam antibiotic therapy more effective than standard of care therapy? So the population was given critically ill receiving beta-lactams. The intervention was TDM-guided adjustment of beta-lactam therapy, and we compared it to the standard of care therapy, which means that the therapy is given like it is in the SPC of the drug. Our primary outcome was clinical cure. Secondary outcomes were microbiological eradication, treatment failure, target attainment, and length of stay and ICU mortality. We conducted our search in three main databases. Firstly, in 2021 November, and then we updated our search. We found 8 articles eligible. I would like to present the results of our investigation. The most important result is that TDM increases the odds of clinical cure compared to standard of care therapy. You can see here that wherever we were able to, we conducted subgroup analysis based on the observational studies and the randomized clinical trials, and then we conducted a pooled effect, which shows that the odds is more than 2 times higher in the TDM-guided group for clinical cure than compared to the standard of care. Treatment failure is the complementary of clinical cure, and you can see here that it's also statistically and also clinically significant that the odds for treatment failure is higher in the standard of care therapy group than to the TDM-guided group. We had the outcome of microbiological eradication. Here we were also able to conduct subgroup analysis, and also the odds for microbiological eradication is almost 2 times higher in the TDM group compared to the standard of care group. We had additional outcomes. One of the most important is the target attainment, which was like, it was not statistically significant, but clinically it's very relevant data. And also mentioning IC mortality and length of stay, which bears not direct relevance to TDM, because these two outcomes are influenced more than only TDM. These results didn't show any statistical significance with the IC mortality and the IC length of stay. The conclusion of this study was that using TDM is beneficial for increasing the clinical and microbiological cure, and using TDM decreases the treatment failure, as I was mentioning before. The population of critically ill patients are very heterogeneous because the diagnosis and the admittance of intensive care unit is very diverse. So we would like to, in the future, homogeneous patient populations and subgroups of antibiotics would be great, because here we weren't able to group the antibiotics like subgroups, so only all the beta-lactams were analyzed together. Standardization of protocols and the clinical endpoints are also essential to create. The implications of this study was for clinically that TDM-guided administration of beta-lactams should be facilitated in intensive care and promoted. And as for research, RCTs are needed with greater sample size and standardization of clinical protocols. And in the future, it would be very nice if we could evaluate the cost effectiveness of TDM, which is quite complicated right now. As for summary, the strengths of our study is that quantitative outcomes were obtained, and we did the highest methodology, what we could do from our articles, and we did subgroup analysis, and the endpoints bear direct relevance to clinical decision making. Also, unfortunately, we had limitations, the small sample size, the risk of bias was moderate, and heterogeneous population. We were able to submit this manuscript yesterday to the Annals of Intensive Care, and we are waiting for the revision or the decision of the journal. I would like to move on with my second project. The focus was on the first project, but as a part of a bigger clinical research group, besides of CTM, I am working with colistin, and we are working to evaluate its clinical impact and pharmacokinetics of this substance. The background of this, that colistin was discovered decades ago, but and then it was abandoned, its use, but now it's also used very frequently on a daily basis because the emergence of the gram-negative superbugs at the healthcare, especially in the critically ill, and we don't know how to promptly guide or dose this substance to the critically ill. Also, as a proof of its crucial importance, I brought some recent publication which shows the incidence of nephrotoxicity associated with the intravenous colistin method. So you can see on this curve that after the 14th days of treatment with colistin, the probability of nephrotoxicity was 50% in patients. So we established a multidisciplinary framework, and we are investigating not only colistin but also beta-lactams. And I think this is the most important message of our work, that the multidisciplinarity is the crucial importance for this work with the laboratory medicine specialist, with the clinical pharmacist and the intensive care clinicians. As a proof of our results, with a bigger working group, we already published into a Hungarian journal, The Challenge in Modern Intensive Care, Chronic Critical Illness. And the part of this bigger project is our projects of TDM, and also the Society of Laboratory Medicine had its conference this year, and the poster was published about our novel in-house method, which was developed for the investigation for colistin. As for overview, I would like to achieve my goals with these two projects. And as for additional activities, here are my posters and the conference presentations which were held last 2 years. Thank you very much.
Thank you, Eszter, for your wonderful presentation. I would like to ask that in your daily practice, how widely available in the clinic TDM-guided pharmacotherapy, and what are the attitudes of the clinicians towards this?
Thank you for your question, Istvan. Yes, we started using or implementing TDM last year, and we have very good results, although there are some challenges. It's always hard to implement or to introduce something new wherever we are. So we are facing difficulties, but now I can say that it's available, not on a routine daily basis, but as for the part of the project, it's available. And as I mentioned, with the intensive care clinicians and with the strong cooperation with the laboratory medicine, we are able to conduct in specific patients. So our aim is to provide it as a routine procedure. That will be the next step.
Thank you very much. I'm Gellert Karvai. I'm the supervisor of Eszter, and I'd just like to make a comment regarding the question of István. We have about 80 to 100 samples coming in for routine antibiotic TDM each month to our laboratory. So this thing has in fact started. Routine TDM of these substances is now available. The TDM of colistin is also available, and we have about 5 clinical departments in Budapest, hospitals and clinical departments, which send us the samples on an everyday basis, and we have a tremendously interesting experience with the results of these measurements. So I think it is wonderful that at the Center of Translational Medicine, the synthesis of knowledge can be performed so efficiently, because at the same time we can see the very everyday practice and the very everyday use of routine antibiotic TDM. Thank you.
Thank you for your presentation. It was, again, really nice. And I, if I understand well, you had to start again your selection. Am I right?
Yes, we had to update our search.
And what was the reason? And do you believe it really improved?
Yes, the reason was not that it was not good before. The reason was that last year, one Australian research group just published the same PICO and Meta. And then when we realized that, we wanted to renew our search to be more updated and up-to-date. And now I think that we made some better investigation. And not going into the details, but the standard of quality is higher. We only selected observational and RCTs, randomized control trials, so only peer-reviewed studies were included in our work.
You mentioned beta-lactams in general. How many drugs were included? And what do you think about there? Is there any difference between them, or we can consider them as a singularity because they are similar in pharmacokinetics meaning? And maybe one more point. What do you think? Is it rational to determine separately the beta-lactamase inhibitor if it is included, or it is not really useful, the beta-lactam antibiotic is good enough?
Thank you very much for your question. I would like to answer the last question. We already measured, for example, piperacillin tazobactam, and as we had our results, the results are the same both for piperacillin and tazobactam. So we concluded that it's useless kind of to measure separately. But as for the first question, it would be very nice because, of course, antibiotics are not the same, varying in half-life, in elimination, half-time. So pharmacokinetics are not the same. but you can see that only 8 eligible studies were found totally for beta-lactam. So we were unfortunately right now unable. So that's why it would be very good to establish a new investigation on this field. I agree, it would be very nice to separately, subgrouply analyze beta-lactams.
And how many compounds did you measure?
We measured the beta-lactam. I mean, in some beta-lactam, piperacin, tazobactam, meropenem were the key substances, but in several studies there were 8, 9 different kinds of beta-lactams, but not all of them included in all 8 studies. Piperacin, tazobactam, and meropenem are the key.
Thank you. In your second publication, you mentioned the method of HBRC combined with MS for colistin. Am I right?
It's LCMS, MS, yes.
LCMS. Yeah. Okay, that's the same. So, is there any reason for abandoning immunological methods? Is colistin a weak immunogenic structure? I mean that chemiluminescence or immunological methods are cheaper than and widely available, not as...
It's not widely available. That's why our colleagues, I'm in the clinic, so I'm more in the clinical part as a clinical pharmacist, but in the laboratory medicine, my colleagues and my supervisor developed an in-house method because it weren't existing before. So it's quite a novelty not only in Hungary, but in a broader spectrum, that we are able to measure colistin in as a terms of TDM.
Okay, I understood. I just addressed the issue. Can you recommend this method for smaller laboratories because it's not widely available?
Yes, I would highly recommend it, but right now the method is just under validation. So what I was showing, this poster, was just a proof of the validation of in-house method and the clinical proof of the validation. So yeah, later on it would be very nice, but I have a doubt that when it would be ready for a wider spectrum, but we are working on that. Very nice.
Thank you very much.
So in your study, you showed an outcome as length of hospital stay, and it's typically given differently in the different studies. So in some cases, they're giving it in with the mean standard deviations. Other cases, they are giving information about it in medians and quartiles. Is it happened in your situation, and how it could be handled?
That it is given as a mean difference?
It's sometimes the length of hospital stay is sometimes given with means,
yes,
and sometimes it's given with medians in the studies, and how you can handle it. It both happens. How you can pull them, or what you should do too.
So it was sometimes it was given mean difference with the interquartiles, and then so in our studies, what we were including, it was given like mean differences. So that's why we pull them together.
As I see, there are stars in some of the studies.
Yes.
What is the meaning for that? Is it not meaning that if there are stars, it was somehow converted from medians to mean?
Yes, they were converted to mean. Yes.
So it was given in medians and quartiles, probably.
So it was given in medians in the articles, and then we were converting it to means.
And another question that what was asked here, that several different compounds, pharmaceutical beta-lactams used in the studies. So is there any idea about that it could give different result in the studies or not? So how you can assess this difference between the studies?
yes, we were facing this problem. We realized that they are different substances, but somehow they belong to the same group of antibiotics. And there are two meta-analyses in this topic dealing with the beta-lactams. They were doing the same method, that they pulled together beta-lactams, because unless we were not able to do that in other way around. So yes, I say that it's problematic, but we could extract data in a consecutive way from each study.
So it means that in a given study, they used always the same, they reported the result from the same beta-lactams, or not?
There were studies where they had only 1 or 2 beta-lactams, and there were studies where there were 5 or 6 investigated beta-lactams. But the results were pulled together also in those articles as well.
And have you seen any difference in the variances? If there are different, if in a study they are included more different beta-lactams, the variance was different?
I cannot really tell that it was different. So
because I think if you see the variances, you use the I-square to show that this is not so heterogeneous. Based on that, you can give a mathematical way to answer the question that is it important to differentiate the beta-lactams or not. I see that it was in the Kaplan-Meier curve.
yes.
It was very strange for me. So it was not your curve. It was in the study.
No, it's just an example I wanted to show you.
Okay, because it's very strange that it will be a horizontal line from a very, very long time. So it means that maybe nobody dies after it, or there was only one person who was observed later.
It was 160 patients were investigated in this study, and the explanation of this figure was like the probability of not gaining nephrotoxicity is 70% in the first 7 days, and the first 14 days it's 50%.
Switching the first.
So I just wanted to show you that it's rapidly.
Yes, and probably who knows what's happening after about 50 days, and it's, I don't think so. This is the best Kaplan-Meier curve to show it is without data after 50 days. Thank you.
Thank you.